Hello, my name's Vrolijk, and this summer I got to look at the difference in community assemblages among interior and edge habitats, as well as between different submerged aquatic vegetation species. So for a little bit of background, the first species we looked at is Vallisneria americana, and it is a native species to the Mobile Tensile Delta. It grows vertically in the water column, and it has ribbon-like leaves. The second species we looked at is the invasive Myrophyllum spicatum, which is known as milfoil, and it creates this sort of dense matting in the water column as well as on top of the water column. So what we wanted to investigate is if that dense matting would create a larger abundance and diversity of species in there, as well as looking at if there's interior and edge effects of species richness and diversity in those habitats. So the way we did that is we went to Chocolate Bay, which is just north of the causeway, and we had 3 treatment groups. We had Vallisneria, milfoil, and bare sediment. For the Vallisneria and the milfoil, we sampled the interior and the edge 10 times. Per sample, we did 1 throat trap to collect nectin, and nectin is basically an organism that can swim on its own against a current, so we were mostly catching fish, crabs, and shrimp. So after those were caught in the throat trap, they were collected, identified, and then released. For epifauna invertebrates, we collected the epifauna from each site, put it in a bag, and brought it back to the lab for identification. So if you look up here at the results, this is our nectin and MDS plot, which basically shows the relationship between all 4 treatments that we had. So this is the Vallisneria interior, the Vallisneria edge, milfoil interior, and milfoil edge. For the nectin, we see the points are sort of jumbled together, no real relationship between that. However, if we go down to the invertebrates, we can see that the milfoil points are all sort of contained to this one area, and the Vallisneria points are all off doing their own thing as well. What does that mean? So basically, that means that the community assemblage in the nectin, there's no difference between any of these treatments. So no difference between SAV type nor interior or edge. However, when we're looking at the invertebrates, there is a difference between the milfoil and the milfoil and the Vallisneria assemblage. However, there is no difference between the interior and the edge. And that same story is being told if we look at our species richness plot. There's no real difference with the nectin. However, if we go down to the invertebrate species richness, we can see that the milfoil supports a much higher species richness than the Vallisneria. So why is that? We think that the structural complexity in milfoil can really harbor a place for these invertebrates to escape predation, whereas the nectin are not as reliant on the structural complexity to escape predation. So in conclusion, SAV type did not affect the nectin at all. However, it did affect the invertebrates. And for all 4 treatments, the interior and the edge did not have any effect on community assemblage. So for the future, I think it'd be cool to sample using an electrofishing boat rather than a throat trap to help capture some of those larger nectin that we couldn't quite trap in the throat trap. Thank you.